Our next speaker is Dr. Tim Banky. Dr. Banky serves as the Pontio Family Endowed Chair in Neurology Research at the University of Colorado in combination with the Children's Hospital Colorado. He is the leader of our Colorado CDKL5 Center of Excellence. But more importantly, he's a doctor who sees value in our kids and loves them.
Good afternoon. My name is Dr. Tim Banky. I'm a scientist and pediatric neurologist at Children's Hospital Colorado, medical director of the Rett Clinic, and lead the site for the IFCR Centers of Excellence. I have personally evaluated nearly 70 patients with CDKL5 Deficiency Disorder, or CDD. These are my disclosures, but none have influenced today's content. So I'm going to give a historical perspective on CDD, review molecular aspects of the cyclin-dependent kinase like 5 gene and protein, discuss the clinical aspects of CDD, and cap with the unmet clinical needs. CDKL5 deficiency disorder, or CDD, is what we call a developmental encephalopathy, which means that the symptoms, not just neurological, change with age. Genetic alterations in the CDKL5 gene were first linked with human disease in 2004 through cohorts of patients seen in Rett clinics that were found to not have alterations in the Rett gene MECP2. These patients were initially ascribed to have a Rett syndrome variant, but more detailed study and experience determined that this really is a unique disease through seminal work by Dr. Leonard's group in 2013. These patient cohorts were assembled and assessed through combinations of smaller case series and family surveys. Recently, we confirmed the unique nature of CDD through our work with the IFCR Centers of Excellence through the use of a uniform clinical data collection instrument in over 92 patients. The CDKL5 protein has 6 major domains. Oh, I'm not sure. So these are the major domains here. And the one here in green is probably the most important. While nonsense or protein truncating changes coded by mutations in the CDKL5 gene are all disease-causing, missense changes coded by mutations associated with the kinase domain are equally disease-causing. Recent studies have determined a birth incidence of disease-causing CDKL5 mutations in 1 in 41,000 births. Prevalence has been more difficult to determine and has everything to do with access to genetic testing. While at least 500 patients are known through Facebook, and we have seen now about 175 in the Centers of Excellence, we estimate from the Colorado experience the prevalence is at least 1 in 75,000 because of our single referral center and our high access to genetic testing. The role and function of the CDKL5 protein is still emerging. It is a kinase localized to the nucleus of all cells and to synapses, the chemical connections between neurons in the brain. Evidence suggests that CDKL5, as a kinase, phosphorylates MECP2 and DNMT1, proteins involved in DNA methylation in the nucleus. And at synapses, it phosphorylates EB2, NGL1, and amphiphysin. These are proteins involved in synaptic structure. At synapses, it also binds and interacts with several synaptic structural elements such as PSD95. Genetic manipulations that knock out or reduce CDKL5 protein in rodent models negatively impact rodent behavior, visual function, and synaptic functions. Despite some limitations, these models support small molecule and large molecule therapeutic approaches. So far, this supports the suggested diagnostic criteria for CDD clinical trials. Broadly, inclusion is any alteration of the CDKL5 gene and confirmation that the alteration is pathogenic. Exclusion would be duplication of the CDKL5 gene. Based on our experience in the IFCR CDD Centers of Excellence, where, as I've said, we've now seen about 175 patients, here is our experience. The disease typically presents with epilepsy beginning as early as 2 weeks of life, which is medically refractory. This often includes infantile and later epileptic spasms that persist throughout life. Interestingly, and consistent with this being a developmental encephalopathy, Many have a seizure honeymoon around 1 to 2 years of age that lasts up to 12 months. With the epilepsy diagnosis and evaluation by a pediatric neurologist, the other impactful features are discovered: cortical visual impairment, severe and global developmental delays, slow development of hand use, and very low muscle tone or hypotonia. Other features emerge later in life, which include movement disorders, severe all-night party sleep disturbances, irregular breathing, swallow dysfunction, severe reflux, and constipation. These features can have different tolls on different parts of the body, such as chronic aspiration pneumonia and scoliosis, often severe enough to require surgical correction. It is important to point out that not everyone is the same and that there is indeed a spectrum of severity. Some, not many, of our patients walk, talk, and have functional hand use. We have found that this is not affected by certain types of or groups of mutations or sex. Epilepsy can, though rarely, improve with age. In our older patients, hypotonia may evolve into dystonia with Parkinsonian features. Some of these I've successfully treated with dopamine replacement. Importantly, there are many non-neurological features that require the attention of pulmonologists, gastroenterologists, and orthopedic specialists. And this leads off our list of unmet needs. Our patients need access to a range of specialists, not just neurologists, but pediatric neurologists and epilepsy specialists, ophthalmologists that recognize cortical visual impairment, pulmonologists, GI specialists, orthopedists that can each manage handicapped children. Experience with CDD is always a plus. Access to both genetic testing and a good genetic counselor to explain the test and the results is absolutely critical to diagnosis. Therapists for physical, occupational, speech, augmentative communication, and educational vision services are required. Because this disease is more than just epilepsy, therapeutics that address intellectual impairment, cortical visual impairment, sleep, movement, GI, swallowing, and medically refractory epilepsy are all needed where none currently exist. In summary, we could not bring this to you without our patients and families with CDD. It's important to recognize their dignity. We ask their forgiveness when we've gotten it wrong, but we recognize their personalities, their teenage eye rolls at their mothers, when they snuggle with their brothers, and the rest of it. We have a great team in Colorado as well as with our partners around the globe that have been able to bring this to you today. So thanks very much.
So we're fortunate enough today to be introducing you to not only one physician that has love and compassion for our children, but two. Our next speaker is Dr. Orrin Devinsky, Professor of Neurology and Neurosurgery and Psychiatry at NYU School of Medicine and leader of our CDKL5 Center of Excellence in New York.
Thank you, Karen, and thanks for the opportunity to speak here and share my views on CDD. To echo what Tim and Karen have said, CDD is a devastating disorder, and it is unique among neurologic conditions. I care for many thousands of children, primarily with epilepsy, and it ranges the gamut of undiagnosed disorders to children who have a gene disorder known in only 1 or 5 other children in the world. But among all those disorders, CDD stands out as unique. So, 2 major problems which you've already heard about, and I think will be the subject of many more presentations today, are severe epilepsy that does not extinguish despite every therapy that's available to modern science today, and global developmental delays that affect every aspect of development: motor function, intellectual function, behavioral function, and physical function. And those are devastating. As Dr. Benke related, there are many other major categories which, depending on the child, can be the paramount and most influential ones that disable the child, but they include GI issues, musculoskeletal issues, visual issues, and autonomic issues. The number of physicians, therapists, nurses, and different healthcare providers involved in the care of people with CDD is at least the 17 professions listed here on the slide. But I must say there are many more that end up getting involved, and I think as the parents know better than either Tim or myself who try to coordinate these, the coordination and integration of care and recommendations with a GI doctor recommending a bowel prep for the child, how does that interact with medications that are given for muscular disorders or epilepsy? They're very complicated, and integrating the care is best done at a center of excellence, but even there, I think we have a long way to go. The epilepsy phenotype, which is my own expertise, is an extraordinarily difficult one to control, and I think we have all, through the period of time we've known what CDD is and treated it, have really thrown at least all 20 of the medications that we have available to treat seizure disorders. We've used neuromodulatory therapies like vagus nerve stimulation. We use dietary therapies. But the reality is none of those therapies has proven close to successful. There are typically the 3 stages. The first one being spasms, beginning very, very early in life, typically one of the earliest presentations of infantile spasms, again, a relatively unique feature of CDD. Then it evolves into an epileptic encephalopathy, where the child then develops cognitive and behavioral delays, and sometimes the sparkle in their eyes slowly starts to fade as seizures and EEG abnormalities become relentless, and neurologists like myself pour more medications on to try to put the seizure fire out. And then finally, they evolve into a treatment-resistant epilepsy, sometimes after a relative honeymoon phase, as Tim mentioned, where seizures come under better control. And then once they come back, they often remain refractory to everything we do. There is one sequence of seizures described in CDD patients which has not been seen with any other genetic or non-genetic pediatric epilepsy. It's a hypermotor seizure with rapid movements followed by stiffening, and then spasms in a very stereotypic sequence. So again, one of the features that points, and I think in some ways confirms, that this gene does cause a unique phenotype in these children, and it's not reasonable to compare it to other genetic disorders or other non-genetic disorders. As I said, for epilepsy, the first thing we do is we use the standard anti-seizure medications. If they have spasms, we'll use ACTH or vigabatrin. After that is a lot of different views based on different doctors, and even within that, because the response rate is so poor, it's really been hard to ferret out, even from large collaborative efforts among the centers of excellence, to find which drugs truly are relatively more effective. But the other side of the equation, as I said, one of the most devastating parts of CDD is the effect on cognitive, motor, and behavior development. And as pediatric epileptologists add more and more drugs at higher and higher doses to extinguish seizures, we often extinguish part of the child's personality and cognition in the process. So we're left with this impossible balancing act between seizures and the side effects of the medications we prescribe. There are other therapies for CDD, but to date we have almost no data to support that any of them are any more effective than the drugs, which are terribly ineffective. What do we do for the most severe problem facing these children, their global developmental delays? Well, certainly we know when seizures are controlled, and many parents can comment on this, relative periods of seizure freedom when drugs are not too high are associated often with cognitive and motor and behavioral gains. The children are more responsive, they're more interactive, there's more sparkle in their eyes, they're more connected. But when seizures come back, those things may be lost. As we go up on the seizure meds, those things may be lost. So currently, the state of medicine is a very bad place for these children. There are many therapies that are beneficial for these children, but in a relatively measured sense. And then there are symptomatic therapies for behavioral disorders, from attentional problems, GI disorders like constipation or lack of GI motility, and many other therapies that can be targeted, but they're symptomatic. And to date, we have no targeted or effective therapies for CDD. So unlike a disorder like tuberous sclerosis, where we have mTOR inhibitors that can literally specifically reverse the abnormality caused by the gene defect, we don't have anything close to that today in CDD. We have no way to modify disease outcome, no way to counteract the underlying problem that affects these children. On a positive note, at least 4 different drug companies have become involved in treating epilepsy in CDD. Ganalaxone is a drug that is currently in a double-blind randomized trial, phase 3. Adalurin is a premature stop codon read-through, small molecule, that's being used to treat 8 patients. That trial is over, and we'll hopefully have a readout on that data in the next month or 2. TAK-935 is another drug with a novel mechanism of action, which is being used to treat children with CDD. And then finally, there's a small open-label 10-patient trial of fenfluramine, which has been a breakthrough therapy for Dravet syndrome and hopefully Lennox-Gastaut syndromes. But all of these are really epilepsy therapies. They're not CDD therapies. They're just other drugs targeting seizures in children with CDD. I hope they're all effective when the data's in, but we'll see. None of them have completed their trials to date. So what are our unmet needs? They're basically everything at this point. We have these anticonvulsants, but it's unclear to what extent they really improve other therapies, and we do a terrible job at seeing how much harm they do. Long-term, these drugs can cause a variety of problems from irritability and aggressiveness, inattention, decreased muscle tone, impaired GI mobility. The list goes on and on. So, we need better drugs. We also need to figure out epilepsy trial designs that look at outcomes beyond seizures. Because if we improve seizures but devastate cognition, I would not call that a good therapy. But right now, understandably, all of the primary outcome measures for these trials are seizure frequency. So we need better criteria to even evaluate the efficacy of the anti-seizure drugs that we're using now. And we need to develop and have ways to measure disease-modifying therapies. One fear I have, which is in some ways a good problem, is from the Dravet experience. For Dravet syndrome, there's now been 2 drugs that I hope, one drug approved, Epidiolex or CBD, and another drug, fenfluramine, that has had exceptionally positive results in randomized trials that I strongly hope the FDA approves when it's presented to them in the coming future. But what that will do is that when there are disease-modifying therapies, as Stoke Therapeutics has an ASO treatment that they're anxious to use in patients with Dravet syndrome, they were always planning on counting seizures. But by the time Epidiolex and Fintepla hit the market, which will be probably well before their trial begins, they won't be able to count seizures so readily because seizures, hopefully, will be relatively infrequent. So in Dravet syndrome, which they're doing, which I think is a great idea, they're looking at other measures of the natural history and how their therapy can intervene. We need to do similar things in CDD so we don't get focused on the epilepsy, and then if epilepsy actually responds well, we may actually have a therapy that reverses CDD, but it will fail in a trial because we don't have an adequate primary outcome measure. So that's another enormous unmet need, that we don't have those measures today. So ultimately, we have many unmet needs. We need to do better at diagnosis, and we need better outcome measures, most importantly, to see how these kids do in their big picture. Some of the potential disease-modifying therapies that are being explored at this time include a protein or enzyme replacement therapy, gene therapy, of which there are a variety of different types, but Ultragenyx and Amicus are leading this, read-through strategies, since about 20% of CDD are due to premature stop codons, small molecules, transfer RNAs, there are a variety of approaches to this, and then potential targets to reactivate X chromosomes or gene editing or antisense oligonucleotides. So I think the future certainly holds a lot of promise, but at the moment I think it speaks to the failure of the medical community as a community to do much to help these children. Thank you very much.